My name is Grace DeRocher, and I am a junior here at SIUC. I've been working in Dr. Shumsey's lab for the last 2 years, and the poster I'm about to present is one of the projects I have had the privilege of working on. I received the REACH Award to gain this research experience, and I presented this project at the Research and Creative Activities Virtual Forum this past spring. Our project proposes a novel peptide nucleic acid probe for the label-free detection of neurodegenerative disease-causing DNA repeats. There are a number of genetic disorders that are caused by trinucleotide expansion repeats, and once the number of these repeats passes its threshold length, it becomes disease-causing. And then the sooner that these disease-causing repeats are detected and these genetic disorders are diagnosed, the sooner treatment can begin, which ultimately leads to a much better outcome for patients. In our project, we test for the trinucleotide sequence CAG, which when repeated 36 to 120 times causes Huntington's disease, which this is a progressive brain disorder and it causes uncontrolled movements, emotional problems, and a loss of thinking ability. Electrochemical techniques can be used to detect these disease-causing repeat sequences. Our experimental scheme, which is shown down here in figure 2, consisted of using a probe strand that contains the sequence CTG, which is complementary to the sequence which causes Huntington's disease. We placed this probe strand on our gold electrode surface and allowed it to attach. Next, we incubated with MCH to cover the unmodified surface and prevent non-specific adsorption. Then we added our target DNA, containing the CAG repeats, which cause Huntington's disease, and allowed these two strands to come together and hybridize, which they should since they're complementary and their base pairs match. Then we ran electrochemical tests such as EIS, or electrochemical impedance spectroscopy, which basically applies a current to the gold surface and gives us a measurement of resistance. The bare gold by itself has an extremely high conductivity and low resistance, but when we place PNA and DNA on the gold surface, it interferes with the electrical current and gives a much higher resistance. This change in resistance tells us that something occurred on the electrode surface, such as the addition of our probe and target strands, or maybe it will even tell us about hybridization. So ultimately, we could use this to determine if a real patient sample of DNA contained these disease-causing repeats based on this change in resistance. However, it was first necessary to create and optimize the biosensing platform, which included our gold electrodes and probe PNA, in order to have good selectivity and sensitivity when testing our target strands. After we optimized the incubation time and concentration of our probe, we moved forward to optimizing our target and then explored the response of different lengths or numbers of trinucleotide repeats of our target. In Table 1 here, you can see the probe and target sequences that we used. Our experimental procedure consisted of cleaning and preparing electrodes, preparing our probe and target strands, incubating with probe PNA, incubating with MCH, incubating with target DNA, and then running EIS tests. We used a PNA probe rather than a DNA probe due to its higher stability. As seen here in Figure 1, DNA contains a lot of negative charges from its phosphodiester backbone. However, PNA doesn't contain these negative charges due to its neutral polyamide backbone, so that makes it a lot more stable than DNA. So first we optimized the incubation of our probe on our electrode surface here. I'm going to talk about this section here. But this allowed it to attach to the gold surface, and then we also optimized the concentration of our probe. We had a range of incubation times from 2 to 20 hours and found that a 4-hour incubation time had the biggest change in resistance, seen here in Figure 3 with this green line. See how it goes out the farthest? That shows that it had the highest resistance. So by the way, this resistance that we're measuring is a diameter of the semicircles that you're seeing here on the graphs. We also tested a range of concentrations for this probe, and we found that a 1 micromolar PNA concentration was our best option as shown here in figure 4, also with this green line with the highest resistance. We then moved on to optimizing our target strand conditions. We selected a half-hour incubation time and a 100 picomolar concentration of our target DNA. So let's talk about figures 5 and 6 here. First, the incubation time. As you can see here, we did not see much difference in resistance as we increased the incubation time past 30 minutes. We did see a jump here from 15 to 30 minutes, but after that there was really no significant change. So we opted to select a 30-minute incubation time just for efficiency. It's much more efficient and saves us time to incubate for only 30 minutes versus 3 hours, especially when this 3-hour incubation time did not give us a significantly higher resistance. Now let's look at the different target concentrations. This is where it kind of starts to get a little wonky. If you notice here, the resistance values after we introduce the target are much lower than they were with just the probe surface. Here they're falling at most around 20,000, so 20,000 or less. While over here you see the probe, it went all the way up to 30,000. Therefore, we can assume that when hybridization occurs, the resistance actually decreases when compared to the probe's resistance. We believe this decrease is due to the negative charges in the DNA interacting with the positive charges of the solution. And the target strand, remember, is DNA, while our probe strand is just PNA. So here we observed that 10 nanomolar, 100 nanomolar, and 1 micromolar target concentrations, they all showed similar resistance values as the probe alone. They showed the highest resistance, which was most similar to the probe's resistance, signaling that less hybridization occurred. We also think that the higher concentration of these values of target saturates the surface, which also contributes to higher signals. On the bar graph here, we see that 1 picomolar and 10 picomolar also show an increased resistance, where as the value increases, hybridization decreases. So we chose this 100 picomolar concentration because this lower resistance told us that it had the most hybridization occurring. Even though this 1 nanomolar here, you might notice it has a little bit of a lower resistance, we still chose this 100 picomolar instead of 1 nanomolar because although it had the lower resistance, it jumps dramatically here when it goes to 10 nanomolar, while there isn't much of an increase around 100 picomolar, so we felt like it was the safer option. Finally, we tested our optimized probe with targets of a variety of repeat lengths, ranging from 6 to 30 repeats, and we found that the highest change in resistance was for this 20 repeat length here. We did see a trend of increasing resistance with increasing length here of up to 20 repeats, but when we got to 30 repeats, the resistance decreased, so that's why we optimized our 20 repeat length here. Overall, our optimized probe and gold electrode could potentially be of use in diagnosing Huntington's disease. However, our results are preliminary and further work is in progress until ready for a peer-reviewed publication. We have plans to continue to test our probe using real sample RNA from patients with this disease to further examine the sensitivity and selectivity of our probe. I am hopeful that these findings will contribute to research on further electrochemical diagnostic methods and be of great use in healthcare in the future. Thank you.